This conference will now be recorded.
Okay, great. So today our speaker will be Dr. David Soucek. He's a research biologist at the USGS Columbia Environmental Research Center. His research has been focused broadly on aquatic toxicology of freshwater organisms, particularly invertebrates, with emphasis on investigating exposure to and the effects of contaminants in aquatic systems, as well as development and improvement of toxicology testing methods for freshwater arthropods, and investigating factors that influence the toxicity of aquatic contaminants. Dave led a research program in ecotoxicology at the Illinois Natural History Survey for 20 years prior to beginning his current position in 2021. So with that, Dave, feel free to get going. And I did put everyone on mute, so you might have to unmute yourself, Dave.
Okay, there we go. Can you hear me now?
Yes, we can hear you.
Sorry about that. And I'm going to share this and switch these. Can everybody see that first slide? Okay.
Yes.
Okay. Okay, well, thank you for the invitation to talk a little bit about what we've been doing at Columbia Environmental Research Center in the area of 6PPD quinone research. Before I move on, I'd like to acknowledge that the work I'm going to show is kind of the work of a bunch of different collaborators. Several different folks here at USGS, Columbia Environmental Research Center, including Rebecca Dorman, James Cantrell, Olivia Watt, Adam Moody, and Jeff Stevens, and then a variety of other. So Rachel Lane is an analytical chemist at Kansas Water Science Center, and then Justin Greer and John Hansen out in Seattle, and then some other academic collaborators, as well as funding from the Environmental Protection Agency and Washington Department of Ecology for further work. So I'm sure at this point most folks have heard of 6-PPD quinone. For those that haven't, it's a derivative of 6-PPD, which is this chemical here on the left. So 6-PPD is a phenylene diamine compound. It's a chemical that's used. It's incorporated into all tire rubber to prevent breakdown of the rubber due to reactions with ozone and other reactive oxygen species. So if this chemical was not in tires, your tires would kind of just dry out and break down quite quickly. So this is really a chemical that's found in all tires. And then under normal driving conditions, tire wear particles come off and are released on the ground, on the highway, near roadsides and so on. And when the 6-PPD-q, when the 6-PPD reacts with ozone in the atmosphere, these two oxygens are added and it becomes this transformation product called 6-PPD quinone. And the reason that this is important is because several years ago, about 4 years ago, it was determined that this paper came out from Jen McIntyre out in Washington, that some compound within tire wear particle leachate is responsible for this, what they call this urban runoff mortality syndrome in Coho salmon. And they found that another species, chum salmon, were not affected by this. But basically, in the Pacific Northwest, in Seattle, in streams, during storm conditions, there would be huge losses of coho salmon. So adults would just suddenly die in a very short period of time, and it typically was correlated with runoff events near roadways. And so they determined that it was a leachate in this tire wear particle, some chemical within that leachate. They then went on to determine that it was a previously unknown chemical. They published this paper in Science, and they determined through a variety of, just a really well done paper, a variety of different experiments, that 6-PPD quinone was the chemical that was responsible, the chemical in this tire wear particle leachate that was responsible for this urban runoff mortality syndrome. And what they found was, so they did some testing and with Coho salmon, and determined that a median lethal concentration. So for those not familiar with ecotoxicology, a median lethal concentration is basically a concentration in an experiment where you calculate that would be expected to kill half of your population with experimental population within a set point of time. So that could be a 24-hour median lethal concentration, or 96-hour, which would be 4 days. Median lethal concentration is often shorthand referred to as LC50, so lethal concentration to 50%. And I'll be using that term a lot during this talk, because that's kind of how we look at short-term acute toxicity for chemicals. But what they found was that the LC50 for 6-PPD in 24 hours was 0.8 of a microgram per liter, so 0.8 of a part per billion, or 800 nanograms per liter, which is a very small amount. They did, and this was with 6-PPD that they were able to extract from tire wear leachate. Once they found this, then other chemical companies were able to make a commercial standard for this chemical, and they retested it, and they found that rather than 800 nanograms per liter, in reality, when you use the commercial standard, 95 nanograms per liter was the amount that was the 24-hour LC50 for Coho salmon. So just to kind of get a rough idea, this is maybe a few drops. Like if you're thinking of like dye in a swimming pool, it's a few drops of dye in an Olympic-sized swimming pool. So it's really a small amount of this stuff. This is parts per trillion, and this is causing rapid mortality within 24 hours for Coho salmon. So this caught the attention of a lot of people, and a lot of other species were tested, including some invertebrates and a few fish, largely salmonids. And to date, there's about 6 species, all salmonids, that have been shown to be sensitive to this. Every other species so far that's been tested for sensitivity to this chemical don't appear to be sensitive up to the solubility limit for this compound, which is about 38 micrograms per liter. So it's really kind of a strange thing. It's really kind of isolated so far to salmonids. So some of the others that have been shown to be sensitive are rainbow trout, brook trout, lake trout, a few others, more recently coastal cutthroat trout, and so on. And so we've learned that a few of these species are sensitive, but we started working with EPA, who was interested. So there's several studies that are demonstrating the sensitivity of these salmonids to the 6-PPD quinone. But when US EPA develops ambient water quality criteria, which are like the concentrations they deem as limits that are safe for aquatic life, they have a certain set of criteria that they require in terms of data quality, and those are outlined in this manual here, this OCSPP 850. And basically, those are things that you look for in a published study that tells you the data quality are good. And so, but a lot of the studies that have been published with some of these fish were didn't meet some of these standards, and this was largely because they were set up to reflect environmentally relevant exposures rather than supporting ambient water quality criteria. So, for example, a 24-hour exposure is not sufficient. You need this. The EPA uses 96-hour exposures for their development of water quality criteria. But in the real world, these things are happening in storm events, and so typically a 24-hour exposure is what's environmentally relevant for these things. Some other issues are excessive loading rates, fish loading rates. So you have too many fish in a container, and again, this is just people who typically aren't designing their studies to be supportive of ambient water quality criteria, and then insufficient analytical chemistry or variable concentration. So the 6-PPD-q, when it's put into water, it tends to in a static. So if you put PPD-q in a beaker, within 24 hours, you'll lose about 80% of the 6PPD-q from solution. So it's an EPA prefers to have good, consistent exposure concentrations over time when they're developing these things. And so they talked to us and to see if we could kind of address some of these issues. And so, and at CERC, we have these what we call proportional diluter systems where we can spike. We can make a stock solution of 6PPD-q. It spikes into this diluter system, and it mixes clean water with water with 6PPD-q in different proportions, and we can do make a 50% dilution series and have that solution renewed up to every 15 minutes. And in this case, we use it up every hour. We add in new water, and so this mixes up the solution. It's then put to these tanks. The fish would be in these smaller tanks, and we can set up this. And so basically, we can renew the solutions of 6PPD-q every hour over the course of a 96-hour exposure. And then so water goes into these cases, and old water comes out this screen, and new water is mixed in, so it keeps the water quality consistently high in terms of oxygen and low ammonia and so on. And so this is what we used. We set up a set of test conditions that would meet these exposures. So again, one of the issues that EPA had with some of the data that were published was that the studies were too short. They were 24 hours. So we did 96-hour exposures, but assessing 24 mortality on the way to that. So we did have 24-hour LC50s as well. One thing we also did was after the 96-hour exposure, we took the chemical away and held any surviving fish for up to a week post-exposure, just to see if there were any latent effects. So that was something that was relatively new, that hadn't been done before. We made sure that the loading was below half a gram of per liter of test solution that passes through in 24 hours, which again meets the standard. These were all done at 12 degrees, which is the acceptable temperature for salmonid species, for most salmonid species. Another thing is that a lot of these tests were done with a variety of different ages, so it was tough to get depending on the fish, and so it's tough to get a good comparison of sensitivity between different fish. So in our case, we use fish that were 3 weeks post swim up, and just for those not familiar with that, these are brook trout eggs that are fertilized, and when they hatch, these brook trout larvae have this big yolk sac, and that over the course of about 2 or 3 weeks, they absorb that yolk sac, and once that happens, they start swimming, and that's the swim up stage. And so for all of our exposures, we use fish that were 3 weeks after this swim up stage, so similar ages. And as you can see, depending on the species that we tested, they were different in size. So coho salmon were much bigger than the brook trout or the rainbow trout. And so to kind of get into the data, on the left, for the next 3 slides, I'll kind of show some data here. But the left panel shows our measured 6PPD-q concentrations over time. And so each line and each color is a separate treatment concentration. And as I mentioned before, if you put these in a static exposure, what you would see is your beginning concentration would drop down to here within about 24 hours. But with our system, with our diluter system that's able to provide new fresh solution every hour, we are able to maintain relatively consistent concentrations for each treatment over the course of 96 hours. And these are average measured concentrations. And then this is a survival plot for again, this is coho salmon. And something that jumps out. So this is time. So we did exposure from time 0 up to 96 hours. And then this is just the number of survivals. So we have your controls and the low concentrations up here. But something that jumps out is that with coho salmon, the mortality occurs very quickly in the high concentration. So within about 3 hours, we started to see mortality. But throughout the test, you can see with this maybe one little exception, almost all the mortality that we observed was within this first 24 hours. So if the fish got beyond that first 24 hours, they really didn't. We didn't really have much more toxicity, and that's kind of reflected in these LC50s. So if at 24 hours the median lethal concentration was 41 nanograms per liter, which is about half of what that first published study where they had 95, but still in the same ballpark. And then if you expose them for an additional 3 days, the amount that it takes to cause half drops down to only 31 nanograms per liter. So a lower LC50 indicates greater toxicity, but this is driven by the fact that we expose them for longer, so the toxicity goes down, and that's probably shown in this dip right here. For brook trout, similar situation with a little bit of a blip here at the end, but really relatively stable concentrations for 6PPD-q. This is a different genus. So Coho Salmon was Oncorhynchus, this is Salmonidais. But again, as we saw with Coho Salmon, very rapid onset of mortality. And in this case, again, with it after 24 hours, really very little change in the amount of mortality in the different concentrations. So in the highest concentration, they're almost all dead within about 3 to 5 hours. And the next highest concentration is 139 nanograms per liter. They're mostly dead by about 10 hours, and then all that were going to died within 24 hours. And so similar pattern, really not much of a difference between. So in this case, very little difference between 24-hour and 96-hour LC50. For brook trout, at 24 hours, less sensitive than Coho Salmon, but again, like the Coho Salmon, not really much difference between the LC50 at 24 hours versus at 4 days. So again, what was going to die was probably going to die within that first 24 hours. The case with rainbow trout, which is the same genus as Coho Salmon, was a little bit different. But, so again, our concentrations were consistent over time. For rainbow trout, much higher. You'll note so with brook trout, our highest concentration was 300, and with Coho Salmon, our highest concentration was 79 nanograms per liter. And these are we base these based on some preliminary experiments where we just did kind of rangefinder tests. But here, our highest concentration over a microgram per liter, over a part per billion, so 1,700 nanograms per liter. But the pattern of survival was a little bit different for this species, so rainbow trout. So while we did see rapid onset of mortality in the highest concentration, we saw some stabilization. But then after 48 hours, we saw that more fish began to die over time. And so whereas for the brook trout and the Coho Salmon, we saw very similar. Almost everything died within 24 hours. The LC50 for 24 hours for rainbow trout was more than double that at 96 hours. So meaning quite a few. If you expose rainbow trout for 4 days, quite a few more will die than will die within 24 hours. And so that was a different pattern, and we don't know exactly why that is, but it could be something about their uptake rates or something like that. That's something that needs to be looked into further. Now I mentioned that we looked at latent effects too, and so this plot could be a little bit confusing because we're already talking about LC50s, and maybe some folks aren't super familiar with that. But what I'm trying to show here is that we have an exposure period from 0 to 24 to 96 hours, and then the time when we take away the chemical up to 7 days later. And what these lines are showing is this LC50 at time X. So for example, at 96 hours, divided by its LC50 at 24 hours. So this gray is rainbow trout, the red is brook trout, and the blue is Coho Salmon. So what this suggests is that what this shows, and it's kind of what I just talked about, at 96 hours, the rainbow trout's LC50 was about half of what its 24-hour LC50 was. And so this is just another way of showing this pattern, but it's also a way of looking at extended mortality over time in this week once we take away the chemical. And so what we did was we exposed them for 4 days, all 3 of these species, and then emptied out the water and quickly gave them fresh water and let any survivors be fed and be held in clean water for a week afterward. And as you can see, we didn't really see any additional mortality. So, you know, and particularly in the case of the Coho Salmon and the Brook Trout, if they get past that first 24 hours, they're basically good to go. But even in the case with rainbow trout, which can endure, you know, greater toxicity if there's exposure is longer, once you take away that chemical, there's really no latent effect, you know, residual effect that's kind of left over. So if the chemical goes away, the fish can survive. This is not to say that they don't have sublethal effects. We didn't look at that, but in terms of lethality, we didn't see any latent effects. And so this is kind of just a busy table, but it shows, so above our 24-hour LC50s for these 3 species, comparing ours in the yellow to previously published reports, and then below are the 96-hour median lethal concentrations, or LC50s, compared to previously published reports. And so there's been a few papers report data for Coho salmon, and we're kind of right in that ballpark. So 41, really similar, actually the same as this low et al. Study. For rainbow trout, we kind of split the middle between these 2 previously published studies. But for brook trout, our LC50 was actually quite a bit lower, and this is probably because of the constant exposure. If you put brook trout in a test with static water and the chemical goes away, you're likely to get a higher LC50 because you don't have as much chemical in there at the end of 24 hours. You know, we had a constant exposure of you know, each treatment had constant exposure, and that's probably why we saw greater toxicity compared to these previously published tests. For 4-day or 96-hour LC50s, our data will be the first published to our knowledge for both brook trout and for Coho salmon. A fair number of studies have been published on rainbow trout, and ours again are kind of on the low end, but in the middle of that study, and that's probably because of the younger age of the organisms we had in that case. So some new data definitely for 96-hour exposures for brook trout and Coho salmon. Moving beyond that, we also tested, you know, there's been a few other species, some cyprinids, zebrafish, you know, a limited number of species outside the family Salmonidae have been tested, but not many. But none have been shown to be sensitive to this chemical so far. We did exposures with largemouth bass and smallmouth bass, again 3 weeks post swim up, we exposed them to a high concentration. So this is higher than you would typically expect to find in a storm event. It's possible it could get that far, but this is pretty high. But again, the solubility limit is only about 10 times that, and we saw no mortality over 2 days at this really high concentration. So just as a reminder, for Coho salmon, the LC50 was 41. Well, the 96-hour LC50 was 31 nanograms per liter. So you know, 2 orders of magnitude lower than this, and they were dying within 24 hours. So really kind of a broad range of sensitivities among fish. So largemouth bass and smallmouth bass not really affected at all at a very high concentration. Some other work that we've been doing here, so in collaboration with folks in our environmental chemistry branch, Adam Moody, who's a research analytical chemist here at CERC, he's developed a tissue analysis method for 6PPD-q. And he uses LC-MS, tandem MS to measure this. And he we recently just had a paper accepted in press for this method. So this will be something that'll be useful going forward. This is published in Environmental Toxicology and Chemistry, and we've got a little bit of data on tissue concentrations. So the panel on the left is tissue concentration in nanograms per kilogram versus water concentration that they were exposed to. And these are some of the fish that we had tested in our some of our mortality experiments. So these are fish that that died, and so we preserved them right immediately after death, and then analyzed their tissues. And so these over here are the Coho salmon, brook trout here, and rainbow trout here. And what you see is in the case of each species, as you increase water concentration, you see an increase in tissue concentration, as you would expect. What's interesting is that the Coho salmon and the brook trout kind of accumulate similar concentrations in their tissues, but at different water concentrations. So you know, these concentrations here for brook trout are much lower in the water. You know, the tissue, the corresponding tissue concentration here is at a much lower water concentration for Coho salmon than it is for brook trout. And then, as you'd expect with rainbow trout, they're exposed to much higher concentrations of water, much higher tissue concentrations. This one on the right shows just a different way of looking at these data. This is bioaccumulation factor, which is basically the tissue concentration divided by the water concentration. And so for Coho salmon, so there's some variability depending on the water concentration, but for Coho salmon and for rainbow trout, the bioaccumulation factor is probably somewhere around 40. So 40, the tissue increases about 40-fold over the water concentration. Whereas for brook trout, again, kind of reflecting how the similar concentration but at higher water concentrations, the bioaccumulation factor for brook trout is less than half of that we saw for rainbow trout and Coho salmon. And why that is, again, we don't know. We need to try to look at some more kinetic type studies, but these were just uptake and not looking at uptake and depuration over time. So that's something that we need to keep looking at. We've done some further studies with a couple different invertebrates. To date, there's been maybe 5 or 6 different invertebrate species that have been tested for sensitivity to 6-PPD quinone. So far, none of them have been found to be sensitive. These are data that, again, in collaboration with folks at McGill University and Trent University up in Canada. We exposed a mayfly that we cultured in the laboratory. This is Neocleon triangulifera, this species shown here. And then these are freshwater mussel juveniles, probably about 7-day-old mussel juveniles. And as you can see here, we exposed mussels to such a high concentration that the 6-PPD-q precipitated. That's what this pink material is kind of getting caught in their mucus trails. But it didn't cause any mortality in these mussels at all. So very extremely high concentration of 6-PPD-q had no effects on either mussels or mayflies. But what was interesting, so 6-PPD-q is only one of many transformation products that come from tire particle leachates. So 6-PPD can, if it goes under ozonation, you get 6-PPD quinone, but it can also go through hydrolysis. And in the water and form a variety of different compounds, including this 4-hydroxydiphenylamine or 4-HDPA. And in our test with this mayfly, we did exposures. We spiked water with 6-PPD-q. We spiked water with 6-PPD, and when we went to measure the 6-PPD in that water analytically, we ended up not finding any 6-PPD, but we found 4-HDPA. So what happens is 6-PPD is added to water and it goes through hydrolysis. It can become a variety of different things, but what we were finding in our solutions was 4-HDPA. So within a short period of time, you add this 6-PPD to water, and this is the ozonation route, so 6-PPD-q. But if you add the parent compound to water, it can become a variety of different things, including 4-HDPA. And these are the measured concentrations of 4-HDPA. We didn't add any 4-HDPA to the water. We added 6-PPD, but it changed to 4-HDPA in this test. And then he saw this mortality of the mayfly. So he thought, well, maybe 4-HDPA is what's causing toxicity for mayflies in some of these solutions. But then when we actually spiked water with 4-HDPA, it turns out that 4-HDPA is also very ephemeral, and when you put it in water within 24 hours, it practically disappears. So we tried taking water and renewing the solutions, adding new 4-HDPA every day, We didn't quite get the toxicity that we observed in this initial test when we added 6-PPD and it turned to 4-HTPA. So, you know, we found some information that different transformation products might be causing toxicity to invertebrates. We couldn't quite nail it down to 4-HTPA, but it could be a whole bunch of different other things as well. So that's some work that'll need to be continued to try to see what other transformation products of 6-PPD might be causing potential toxicity to species. So just to kind of conclude, just to kind of recap, so our pulse flow diluter system was really able to produce consistent 6-PPD exposure concentrations with test conditions meeting these US EPA requirements for ambient water quality criteria. And our 24-hour 6PPD-q LC50s for coho salmon and rainbow trout were similar to previously published reports, but for brook trout we were quite a bit lower, and that again is probably because of this consistent exposure concentration. Two centrarix, smallmouth bass and largemouth bass, were not sensitive at all at really a high concentration of 6-PPD-q. Going back to the kind of the kinetics, the coho salmon and brook trout, the 24-hour and 96-hour LC50s were really similar, but for rainbow trout, we really saw a lot more mortality over the course of that next 3 days. And so there's these differences among species, even within the same genus. So coho salmon and rainbow trout are the same genus, just different species, but there's really a lot of variability in how this affects species. Oops. And so after 4 days exposure, and then continued maintenance, if you take the chemical away, continue the maintenance in clean water, the maintenance of the surviving fish, we observed no latent mortality. And then we observed concentration-dependent bioaccumulation in fish tissue with some differences in uptake and in bioaccumulation factors among these different species. And invertebrates were not sensitive to 6-PPD-q, but are probably sensitive to a variety of different other transformation products. So where we're going next with this, in the next week, we're going to start tests with another species of salmonid, so Arctic grayling, so Thymalis arcticus. So far, all the species that have been shown to be sensitive to 6-PPD-q are in the genus Salvelinus and Oncorhynchus. This will be a different salmonid species in a different genus, kind of a different subfamily as well, but again, a salmonid. So it'll be interesting to see whether or not these Arctic grayling are sensitive, and we're going to test them again at 3 weeks post swim up. So that'll start pretty soon. And then we're funded by Washington Department of Ecology to investigate alternatives to 6-PPD-q. So they're very interested in, like, if we use other similar chemicals to stabilize tires, do the transformation products and do these other tire stabilizers have different effects on different species? And our part is going to be looking at freshwater mussels. So if the 6-PPD alternative is less toxic to coho salmon, is it more toxic to a mussel? And so that's some work we're going to try to determine that this summer. With that, I'll just acknowledge some funding from the USGS Environmental Health Program within the Ecosystems Mission Area and the US EPA's Office of Water, and I'm happy to take any questions folks might have.
All right, thanks, Dave. I'm going to stop the recording and then we can move on to questions.